Hello everyone. Our lesson for today is about DNA replication during the S phase. So in this lesson, we will discuss the events that occur during the synthesis of the DNA and the S phase of the cell cycle. The first part will give a review of the central dogma of molecular biology. Then, the molecular structures and functions of the components of DNA replication will be focused on. Main reference for discussion is the book that is written by Watson et al. In 2014. So at the end of this lesson, you will be able to review the central dogma of molecular biology, determine the functions of the different components of DNA replication process, discuss the structure and catalysis of DNA polymerase, and discuss the exonuclease proofreading activity of the DNA polymerase. Before we go into our discussion proper, let us have a quick review first on your knowledge of DNA replication. So, in your past subjects, you have already learned how to make a complementary copy of a DNA strand. Okay, so we know that the base pairing of the two strands of a DNA is complementary to each other. Okay, so in this given DNA strand, you provide the complementary copy that is based on Watson and Crick base pairing. So you can write your answer as like this. Okay, are you finished? So complementary base pairing: T pairs with blank, A pairs with blank, C pairs with blank, and G pairs with blank. So what is the complementary strand of the given strand? And you're correct. Now the complementary strand in the provided DNA strand is ATG, CGAAT, GGT, CTA, CG, AA. No? So, and the direction is from 5 prime to 3 prime. So the question is, how does the cell able to synthesize this complementary DNA strand from this? So this lesson, we will be talking about the molecular details of how one strand is synthesized using another strand as a template. Okay, so the general pathway for the flow of genetic information, known as the central dogma of molecular biology, was originally proposed by Francis Crick in 1956. And this indicates that the genetic information flows from the nucleotide sequence of genes to the so if this is the nucleotide sequence of a gene comprising of the DNA, okay, that codes for the structure of the protein. Now, generally, the DNA is the long-term storage molecule of hereditary information that determines the development, structure, function, and reproduction of an organism. So all modern cells have DNA genomes. However, some viruses now have RNA genomes, and RNA can therefore also be used as a genetic carrier. Okay, so in a process called replication, DNA is copied into more DNA copies and passed on from one generation to another. DNA will also be expressed into its functional products, proteins, and functional RNA. So these functional products constitute the machinery of a cell and perform specific tasks. So from DNA, you can produce an RNA through the process of transcription And that RNA is then expressed into protein through the process of translation. So this view of genetic information is still valid to a large extent today. However, if we could use no, uh, if we could use RNA as a template to produce a DNA copy in a process called reverse transcription, proteins, on the other hand, never serve as a template for the synthesis of RNA. Okay, so the complete reversal of this proposed genetic flow is impossible as of the knowledge today. So maybe one of you will be able to prove this isn't right and will be able to synthesize RNA from protein sequences. Okay, so RNA genomes from retroviruses and are converted into other RNA copies as well through RNA replication. Okay, others, as we have said earlier, through the use of uh, reverse transcriptase, RNA could be used as a template to produce more DNA. Okay, so we could say that DNA and RNA could be used as hereditary agents. Okay, so RNA in some viruses, they are used as their hereditary agents. So in general, two classes of macromolecules are among the major players in molecular biology. You have the proteins and the nucleic acids, the DNA or the RNA. So as we discussed in a previous lesson, these molecules are composed of amino acids that make up the protein and nucleotides that make up the nucleic acid. These are linked by covalent bonds in peptide chain, so peptide bond, and polynucleotides via phosphodiester bond. Now, DNA replication, okay, is semi-conservative. What does it mean, semi-conservative? So the self-complementary structure of the double-stranded DNA helix based on the formation of AT and GC pairs, just like what we did in the motivation part, the first part of our discussion, suggests that each strand serve as a template for the formation of a complementary daughter strand. Hence, a DNA molecule might be copied with retention of the genetic information, thereby generating two identical daughter molecules that's starting with a single template molecule. So in simple terms, one strand provides a template for the synthesis of a new strand, so that at the end, when the two DNA molecules are formed, each copy of the DNA has an old strand and a new strand. So only one strand is conserved for each copy. That's why it's called semi-conservative. Now, what are needed in order for a DNA to be synthesized? So in order for the DNA to be synthesized, okay, so enzymes are needed. So enzymatic synthesis of DNA requires two essential substrates. So what are these substrates? The 4-deoxynucleoside triphosphate, such as DATP, DGTP, DCTP, and DTTP. Okay, so DNTPs, and a primer template junction. So this should be this is one name, okay, primer template junction. So the template junction should be bold and italicized as well. So you have here the structure of a deoxynucleotide triphosphates, deoxynucleoside triphosphate. Okay, so you have here a nucleoside that is composed of the sugar and the base. A nucleotide is composed of one phosphate attached to a nucleoside, and a nucleoside a diphosphate, two phosphate groups attached, and three phosphate group is the DNTPs, nucleonucleotide phosphates. They have three phosphoryl groups: the alpha, no, proximal position, the beta, and the gamma, that is attached to the 2' deoxyribose via the 5' hydroxyl, no, structure. Now, the prime here refers to the position of the carbon, so that they will be distinct from the carbon that is found in the base. You should remember that. So that in the strand of a DNA, the 5' no, refers to the free 5' carbon in at one end of the DNA strand, and the 3' meaning there is a free unbounded carbon, no, at the other end of the DNA strand. Okay, so the beta and the gamma bonds are high energy. So this is alpha, beta, and the gamma bonds. So these are high energy bonds. The primer template junction now has already two essential components. First is a single-stranded DNA template that will direct the addition of the complementary nucleoside to the growing chain. The second is a short primer that is complementary to the template. So this primer consists of DNA and not RNA and not DNA. So they are here. So the components of a primer template junction is the single-stranded DNA template. Okay. This is the one that directs the addition of the complementary strands. So for example, here the C, so the complementary base pair should be G added. Okay, and the second is a short primer. Okay, that is a complementary also to the template. Now this primer, no, is not a DNA strand, but an RNA strand. This is important because this will provide the 3' OH end, no, to where the first DNA nucleotide will be attached. Without primer, no, an RNA polymerase, an enzyme, cannot add the DNA nucleotides in here. That's why, in order to prime the reaction, an RNA primer must be added first, no, by the RNA polymerase. So DNA is synthesized by extending the 3' to 5'. Okay, so the direction of DNA synthesis is from 3' to 5'. Okay, that is the direction. So the strand to be synthesized is from 5' to 3', because the direction is at the 3'. Nakuha? So hydrolysis, how is this possible? So you have here the catalysis of the reaction. So once the primer is already added, and it provides the 3' OHN, a nucleotide, no, a DNTP, no, will then be added. So in this case here, this is thymine, so the DNTP should be a DATP, no? And what happens is that the OH here, the oxygen, will have a nucleophilic attack attacking the alpha position of the phosphate. Okay, releasing pyrophosphate in the process. So pyrophosphate is meaning two phosphate groups that are released. Now this here is highly unstable. It needs a lot of energy. That's why an enzyme called pyrophosphatase will cleave the bond between these two phosphate groups, releasing energy that could be used to stabilize the bond between these nucleotide to the nucleotide that was just added. Okay, so this is what we call the DNA synthesis. This is how they add the nucleotides, how the DNA strand grows. So what are the different components of the DNA replication? So they need a DNA helicase, The helicase is the one that unwinds the double-stranded DNA. Then the DNA polymerase, the one that adds the DNA nucleotide. There are also single-strand binding proteins that will protect the exposed single strands of the DNA because that is prone to degradation. So they need to be coated with this SSB or single-strand binding proteins. Okay. Now, because of the nature of this reaction, sliding clamps are also used in order to push the polymerase along the exposed template so that it will add DNA nucleotides more efficiently. So these are the major components of the DNA replication process. So let us focus first more on the let us take a look at the structure of a DNA polymerase. Now, so DNA polymerase, also RNA polymerase, is an unusual enzyme because it uses only a single active site to catalyze the addition of any of the 4 DNTPs. So if we compare that to other enzyme, one substrate is only catalyzed by only specific enzyme. But this time, for DNA polymerase, it could catalyze the addition of any of the 4 DNTPs. So the DNA polymerase can do this by exploiting the nearly identical geometry of the AT and GC base pairs. Therefore, the DNA polymerase monitors the ability of the nucleotide to form a correct pair with the template rather than detecting the exact nucleotide that enters the active site. So it does not really detect the structure, but how the fit of the pairing between the template and the incoming nucleotide. Only when a correct pair is formed that the 3' OH of the primer and the alpha phosphate of the incoming nucleotide will be in the optimum position for catalysis to occur. Okay? So thus the phosphodiester bond is formed. So unlike this, so you have here G. Okay? So it must be paired with C. Okay? So this should be there at the optimal position for the catalysis to occur. Incorrect base pairing leads to dramatically lower rates of nucleotide addition due to catalytically unfavorable alignment of the substrate. Now, DNA polymerases, they show high discrimination between ribo and deoxyribonucleoside triphosphate. So the discrimination is mediated by a steric exclusion of the RNTPs from the active site. So the binding pocket is too small to accommodate the extra 2' OH group on the sugar moiety of the incoming nucleotide. So we know that a deoxyribonucleotide has no OH group at the 2' carbon of the sugar. But the RNTPs, the one in RNA, have. So if RNA is the one that is incorporated, it will form a steric clash with the discriminator amino acids in the binding pocket of the RNA polymerase. And so it will not create a perfect position for the catalysis of the 3' OH and the alpha phosphate of the to create the phosphodiester bond. Okay. So DNA polymerase, they act to extend the primers that are generated by the primase. So these enzymes are divided into 6 families based on phylogenetic relationships. However, not all of these are replicative polymerases. Some are utilized solely for DNA repair. So only replicative, those that are involved in DNA replication, share common structural aspects. So structural studies of the DNA polymerase Another polymerase is bound to the primer template junction has revealed that the DNA substrate sits in a large cleft that resembles a closed right hand. Huh? So based on this analogy, the three major domains of the enzyme are called the thumb, the fingers, and the palm. Now, how do these three domains differ? So the palm domain provides the catalytic active site due to the important conserved sequence motifs that it contains. So it is accomplished by this task by binding two divalent metal ions, the magnesium 2 plus or the zinc 2 plus, that is held in place by the interaction by two highly conserved aspartate residues. So you have here a metal ion A and a metal ion B. So the metal ions, they alter the chemical environment around the correctly base pair DNTP and the 3' OH of the primer. So one metal ion primarily interacts with the 3' OH, resulting in a reduced association of the oxygen and the hydrogen atoms, thereby leaving a nucleophilic 3' oxygen. The other metal ion interacts with the triphosphates, okay, the triphosphates of the incoming nucleotide to neutralize the negative charge of the phosphate group. After catalysis, the pyrophosphate product is stabilized through similar interaction with the second metal ion. Okay? In addition to its role in catalysis, the pump also monitors the accuracy of the base pairing for the most recently added nucleotide. The fingers, on the other hand, help in the positioning of the template in the active site correctly. So it associates with the template. So this interaction results in the nearly 90 degrees turn of the phosphodiester backbone of the template immediately after the active site. So the bend will expose the first template base after the primer at the catalytic site and avoids any confusion concerning which template base is ready to pair. So one of the helices of the finger domain, called the O helix, okay, rotates by 40 degrees when the correct base pair has been formed. So the polymerase then grips the template and the incoming DNTP when the correct pair is made. In the open conformation, the O helix is distant from the incoming DNTP, but in the closed conformation, this helix moves and makes several important interactions with the incoming DNTP. So a tyrosine residue here makes stacking interactions with the base pair of the DNTP, and the two charged residues, the arginine and the lysine, will interact with the phosphate. So the combination of these interactions together with the interactions from the previously we discussed in the palm domain, will involve in the interaction positions the DNTP for the catalysis that is mediated by the metal ions bound to the polymerase. So the formation of the phosphodiester bond leads to the reopening of the fingers and the movement of the primer template junction by one base pair. So then the next DNTP can then take it up and the whole cycle can be repeated again. Now the thumb domain is not directly involved in the catalysis, but the thumb interacts with the DNA that has been recently synthesized and leaves the enzyme. So these interactions are mainly electrostatic interactions between the phosphate backbone and the thumb domain. So this ensures a strong interaction between the enzyme and the substrate. So the tight association contributes to the ability of the DNA polymerase to add many DNTPs each time it binds a primer template junction. So in other words, DNA polymerase is a processive enzyme. So processivity, we are referring to it as a total number of DNTPs that you added in a particular time. So a highly processive DNA polymerase, of course, can add more nucleotides to a growing DNA strand. Okay, so the higher the processivity, the faster the synthesis. So most of these highly processive DNA polymerases are the replicative polymerases because they are the ones directly involved in DNA replication. Those that are involved in DNA repair are not that processive. Okay, so having said all of those things, how does the process of DNA replication occur? So you've already known before in the past in your past subject that two strands no are formed. So lagging strand and the leading strand. The leading strand no is synthesized continuously now because the template strand is already 3 prime to 5 prime, and so the synthesis will be 5 prime to 3 prime. You just add the primer and the synthesis will go on continuously, so it is a leading strand. Lagging strand, on the other hand, is synthesized discontinuously. That's why DNA replication is called semi-discontinuous, because one of the strand is synthesized discontinuously, and this strand is called the lagging strand because it uses the template, which is 5 prime to 3 prime. And since we know that the DNA strands are anti-parallel to each other, the strand that must be synthesized is 3 prime to 5 prime. Since 5 prime ang free carbon, then it cannot continuously replicate. Okay? Thus, it needs to open up first a certain portion of the DNA before a primer can be added, and the synthesis could then proceed from 5 prime to 3 prime. Okay? So this is a discontinuous synthesis. So what happens? So this is the lagging strand. Okay, this is the leading strand synthesis. So why is DNA anti-parallel? So that it could create a complementary complication for the simultaneous replication of the two template strands, so that the two strands could be simultaneously replicated. Now, because they are anti-parallel to each other. Okay. So what happens next? Okay, so in the lagging strand, RNA primers are added by the RNA polymerase or the primase. Okay, then from there the new DNA is synthesized. This creates an Okazaki fragment, RNA primer plus a fragment of the DNA. The Okazaki fragments are then the RNA primers are then removed, and the DNA fragments are then ligated via the enzyme ligase. Okay. So how are these removed? So an enzyme called RNase H removes the RNA primer. Then the 5' oxynuclease activity of polymerase, not really a replicative polymerase, will remove the remaining ribonucleotide that is attached to the deoxynucleotide, and then a DNA polymerase will also add the missing nucleotides here to seal the gap, and then a ligase will provide the catalysis for the synthesis of the phosphorester bond between the fragments. Now, how do the enzyme make sure that it is free of any mistakes? Because it is highly precessive, it could add hundreds in a second. So how does it make sure that it is free of any errors? Okay, so DNA polymerase have proofreading activity, so it can proofread. It can check whether may mali ba siya or wala. So it has a proofreading nuclease activity which are capable of degrading DNA starting from a 3' end of a growing strand. So we call it the exonuclease activity. So it gives the synthesis activity of the DNA polymerase a second chance to add correct nucleotide. So the removal of the mismatched nucleotide is facilitated by the fact that the DNA polymerase has a reduced capacity to add a nucleotide adjacent to an incorrectly base paired primer. So it will lose its processivity in the process because of structural distortions and poor interaction. So it could sense something must be really wrong. And so when a mismatched base is present in the active site, the primary template junction is destabilized, creating a 3 to 4 base pairs long stretch of unpaired DNA because the nuclease activity has a tenfold higher affinity for single-stranded 3' ends. The newly unpaired 3' end moves from the polymerase active site to the exonuclease active site where the incorrect nucleotide is removed. So the primer template can then reform and rebind the polymerase active site, allowing synthesis to proceed further. So the exonucleate oxidase active site may be present on another subunit of DNA polymerase. No, so DNA polymerase for eukaryotes and DNA and pol1 for the prokaryotes. Okay, so those are the different concepts that you have to learn regarding our lesson for today on DNA replication during the S phase of the cell cycle. So in order for you to review this, I have given summary questions. So what are these? What is the central dogma of molecular biology all about? What are the different components of DNA replication? What are their functions? How does the DNA polymerase work? What is the overall process of DNA replication? And how does the DNA polymerase correct mispaired bases? So I hope you will be able to answer this one as you go along the lesson that we have discussed. I would like to thank again Mr. Ern Oliver C. Balondo for the help in making the slide presentation. So if you have any question, you are free to contact your lecture instructor via email, or you can key in your questions to the official Facebook Messenger group chat. Thank you very much, and see you again in our next lesson.